Hi everyone, welcome to today's webinar entitled Wake Up and Smell the Chromatin, Long Non-Coding RNAs and Development of Disease Progression. My name is Ed Davis. I'm the Technical Support Specialist for Arraystar LncRNA in Rockville, Maryland, and I want to thank you for joining me for today's webinar. We really appreciate it. Today I'm going to talk about an exciting new class of molecules that have emerged recently as important factors in the regulation of gene expression. Long non-coding RNAs, also known as LncRNAs. Before I get started, I just want to take care of a few practical matters. First, you may ask questions at any time. However, you'll not be able to speak vocally to me. Instead, you'll need to type your questions in the questions field in your control panel. Also, in the interest of keeping the flow of the talk going smoothly, I'll wait until the end of the talk to answer your questions, at which time I'll answer each question in the order it was asked. I'll read each question out loud before answering, and then I'll answer the questions so that everyone can hear. If you cannot see your control panel, it might be hidden. In that case, look for a small strip of buttons on the right-hand side of your screen. The top button will be orange with a white arrow. Clicking on that arrow will reveal the control panel, and then you'll be able to type your questions. Okay, so let's get started. Here's an outline of today's seminar. First, I'll give you an introduction to what LncRNAs are and how they're expressed. Next, I'll go over the biological significance of LncRNAs and how they function in some normal biological processes. Next, I'll tell you about how LncRNA misexpression can contribute to disease in humans and how LncRNA profiles can be used as biomarkers for disease. Then finally, I'll tell you about the challenges that need to be overcome in order to do genome-wide profiling on LncRNAs and about technologies developed by Arraystar for profiling this intriguing family of transcripts. So now for part one, the introduction to LncRNAs. Despite the importance of protein coding genes in our lives, only 1 to 2% of the human genome encodes proteins. However, about 75% of the genome can be transcribed among different cell types. This body of non-coding transcripts is comprised mainly of ribosomal RNA, transfer RNAs, microarrays, Pee-wee-associated or Pee-wee-interacting RNAs, and the subject of today's talk, LncRNAs. So again, the take-home message is that most of the human genome is transcribed, but very little of it encodes proteins. Biologists are familiar with the classic central dogma in which DNA makes RNA makes protein. However, we now know that non-coding RNAs can activate and repress transcription and activate or repress translation, and so have major influences on the processes of transcription, post-transcriptional modification, and translation. Long non-coding RNAs, dismissed for decades as either pseudogenes or transcriptional noise, are the latest members of this mysterious group of regulatory RNAs to be studied. So what are LncRNAs? LncRNAs are defined as eukaryotic RNA molecules greater than 200 nucleotides in length, a size arbitrarily chosen based on filtration columns that exclude species smaller than that size. In addition, LncRNAs appear to have no protein coding capacity. Intriguingly, there are about the same number of LncRNAs in mammals, in mammalian genomes, on the order of 20,000 or so, as there are messenger RNAs. Further, LncRNAs are involved in normal development, they're known to regulate gene expression at many levels, and their inappropriate expression can cause disease. The expression and structure of LncRNAs is very similar to that of protein-coding messenger RNAs. As seen in this table, both are transcribed by RNA polymerase II, both can be spliced from larger intron-containing precursors, both are often polyadenylated, and both contain 7-methylguanosine caps. The biggest difference between LncRNAs and messenger RNAs, however, is that LncRNAs do not appear to be translated into proteins. Another fundamental difference between LncRNAs and messenger RNAs is that while mRNAs, represented in green in this figure, are nearly always exported to the cytoplasm for translation into proteins, LncRNAs, represented in black, almost always remain in the nucleus. This phenomenon is consistent with the observed role that some LncRNAs play in the transcriptional and post-transcriptional regulation of gene expression. Now I want to shift the attention of my talk to the expression of LncRNA genes. LncRNAs are transcribed in many different ways. Some, shown in purple, are known to occur intragenically from within genes via alternative splicing of protein-coding messenger RNAs. Intragenic LncRNAs can be transcribed antisense to mRNAs as well, as shown in red. Finally, the expression of some LncRNAs occurs without overlapping protein-coding genes at all. This latter class, shown in green, is known as large intergenic noncoding RNAs, or LncRNAs. As I mentioned at the beginning of the talk, LncRNAs are known to be involved in many, if not all, of the steps in the expression of protein-coding genes. Here are a few examples depicted in the figure. LncRNAs, if expressed just upstream of an mRNA promoter, can cause transcriptional interference with that promoter. LncRNAs can also induce chromatin remodeling via either histone modifications or cytosine methylation. Taking this a step further, LncRNAs can have influence on alternative exon splicing, shown here, or if expressed antisense to an exon, can cause the generation of interfering siRNAs. These are just a few of the fundamental mechanisms known to be affected by LncRNAs, but there are certain to be many more. LncRNAs tend to be expressed with tissue and/or developmental stage specificity. This tells us that the expression of the genes encoding many LncRNAs must be tightly regulated. As I mentioned earlier, LncRNAs share many of the same characteristics as their protein-coding messenger RNA counterparts. Since LncRNAs are also transcribed by RNA polymerase II, their promoters are regulated by the same mechanisms as well. For example, LncRNAs are regulated by well-characterized transcription factors. In one study, John Rin's group at MIT showed that the well-known tumor suppressor protein p53 binds to the promoter of an LncRNA, LncRNA p21, and induces its expression in response to DNA damage. LncRNA p21 is required to activate the DNA damage-dependent apoptotic function of p53. In another example, Leonard Lipovich's lab at Wayne State University demonstrated that two LncRNAs are regulated by the transcription factors OCT4 and NANOG during the differentiation of mouse embryonic stem cells. In addition, LncRNAs are subject to control by epigenetic chromatin modifications. In a collaboration between John Rin's and Eric Lander's labs, it was shown that many LncRNAs carry a chromatin signature, histone H3 lysine 4 trimethylation in promoter regions, and histone H3 lysine 36 trimethylation in the body of the gene. These so-called K4, K36 domains are found in protein-coding genes too, and so are excellent predictors of the occurrence of transcribed units. Trimethylation of histone H3 and lysine 4 and lysine 36 tend to be a mark for activating gene expression in so-called open chromatin. By contrast, LncRNAs genes, like protein-coding units, become methylated at cytosine residues, most notably at CpG dinucleotides. CpG methylation, primarily in promoter regions, is a well-known epigenetic modification that tends to repress gene expression in so-called closed chromatin or heterochromatin. Okay, so now I'd like to move on to part 2, the biological significance of LncRNAs. Sorry, I'm having some technical difficulty here. Be with you in a moment. Okay. So, several LncRNAs play critical roles in well-known biological phenomena. One example is hot air. Hot air is a 2.2 kb LncRNA expressed from the HOX-C locus. Hot air interacts with the polychrome repressor complex 2, or PRC2, to remodel chromatin and repress gene expression at the HOX-D locus. In addition, the most famous of the LncRNAs is Xist, which has been known as a non-coding RNA since its discovery by the Willard Lab in 1992. Xist is a 19 kb long non-coding RNA expressed from inactive X chromosomes in female mammals, and so, along with its antisense partner T6, is essential for X chromosome dosage compensation. To further expand on the example of Xist, shown in this slide is a fluorescence in situ hybridization, or FISH experiment, in which a fluorescently labeled Xist probe, in green, associates with only the inactive X chromosome in an interphase mouse nucleus. The chromosome DNA, in blue, is stained with DAPI. Again, this is a perfect example of an RNA transcript that remains in the nucleus. Okay, so now it's time for part 3, LncRNAs and disease. LncRNAs play critical roles in the biology and development of mammals, so their inappropriate expression might be expected to cause disease. Therefore, if a disease state is caused by the misexpression of an LncRNA, that LncRNA could potentially serve as either a diagnostic marker or treatment target for that disease. In one landmark study, Carlo Croce's lab at Ohio State University examined the expression patterns of long non-coding RNAs in cancer. Using a custom microarray followed by hierarchical clustering analysis, they found that many up and down regulated LncRNAs in at least one type of cancer. The results shown in this table suggest that each cancer carries a signature of LncRNA expression, and that LncRNAs offer promise as biomarkers used for the diagnosis of cancer. So as you can see here in CLL, chronic lymphocytic leukemia, they found a total of 19 differentially expressed LncRNAs, 8 of those upregulated and 11 downregulated. Similar result was found for colorectal carcinoma, 61 total differentially expressed LncRNAs, and finally in hepatocellular carcinoma, they found a total of 8 differentially expressed LncRNAs. In addition, Croce's group found a list of 5 LncRNAs, the expression of which negatively correlates with that of several microRNAs that are aberrantly expressed in chronic lymphocytic leukemia. These microRNAs, and by extension their LncRNA counterparts, can distinguish between 2 classes of CLL based on the expression pattern of the zinc finger transcription factor ZAP70. Earlier, Croce's group showed that elevated expression of ZAP70 in CLL patients is associated with poor prognosis. So not only can LncRNA expression be used to determine the type of cancer a patient presents with, but these results suggest that LncRNAs can be used to predict a patient's survivability of a particular cancer as well. As a further indicator of the relevance of LncRNAs to cancer biology, Croce's lab focused on one particular LncRNA, UC.73, which is upregulated in the colorectal cancer cell line. Knockdown of UC73 by siRNA inhibited proliferation, shown here, of these cells, and in addition caused an increase in the number of apoptotic cells, shown on this side. These results suggest that an LncRNA can act as an oncogene or as a mediator of oncogenic action. Another example of an LncRNA acting as an oncogene comes from Howard Chang's lab at Stanford University. In an examination of hot air, which, as I mentioned before, modulates chromatin structure at the Hox-D locus, Chang's group overexpressed hot air in 4 different breast carcinoma cell lines. As shown on the left, in each of the 4 lines, overexpression of hot air enhanced growth on soft agar, an assay for invasiveness and metastasis. You have in the red bar shown here. Further, in the MCF7 cell line, siRNAs directed against hot air reduced cell growth on soft agar, shown on the right-hand side. Together, these results suggest that abnormal expression of hot air can enhance the progression of breast cancer to a more invasive malignant stage. Another notable disease that's been linked to the misexpression of an LncRNA is Alzheimer's. The Volstead lab at Scripps found that a 2 kb LncRNA antisense to the beta secretase gene, shown here, base 1 gene, is induced in response to numerous cell stressors, including serum starvation, neurodegeneration, and A-beta peptides. Fagihi et al. Proposed that base 1AS, which is this antisense LncRNA, stabilizes base 1 mRNA, leading to the production of even more A-beta peptides and contributing to the worsening pathology of Alzheimer's patients. However, treatment with siRNAs against base 1AS reduces the level of A-beta peptides, offering potential promise for using base 1AS as a treatment target for this devastating disease. So that concludes the introductory part of my talk. What I've told you about so far is what LncRNAs are and how they're expressed, the biological functions in mammals and their roles in disease initiation and progression. Now I'm going to tell you how we can use LncRNAs, in biological investigations and how we profile them. So what are the types of applications for LncRNA analysis? This slide shows some but not all of the most important types of investigations scientists can undertake using LncRNAs. First, one can examine changes in LncRNA expression during development, say between pluripotent stem cells and differentiated tissue. Second, LncRNA expression can be profiled in various disease states, for example, between normal liver and hepatocellular cancer tissue, or among different stages in liver cancer progression. Third, you could examine changes in LncRNA expression following treatments with various drugs or drug candidates. Fourth, you can follow changes in LncRNA expression in response to exposure to various toxic or non-toxic environmental agents, such as pollutants. These are just a few examples of the types of projects investigators can apply LncRNAs to. There are certain to be many others. Okay, so now you're all excited to profile the expression of LncRNAs in your favorite developmental or disease model. How do you go about doing this? Well, the availability of complete genome sequencing has led to an explosion in the analysis of annotation of transcribed regions of the genome, including the identification of LncRNAs. Currently, though, there are a number of hurdles to overcome. First, there's no single comprehensive database of all the known LncRNAs in human, mouse, rat, or other organisms. Second, it has proven extremely difficult for various reasons to design LncRNA probes. And finally, the low expression level of LncRNAs, which is typically lower than messenger RNAs, has lent additional difficulties to studying LncRNAs. Now, Arraystar has three custom designed solutions for LncRNA analysis. The first is the LncRNA expression microarrays, which profile the expression of all the currently known LncRNA genes in human, mouse, and rat. The second LncRNA solution is the LncRNA promoter microarray, which profiles the DNA promoters of all the known LncRNA genes in humans, as well as other interesting genetic elements that I'm going to discuss later. And third, we have recently developed and introduced microarray that profiles a class of elements called TUCRs, which is a specialist subclass of long non-coding RNAs that are highly conserved among various organisms. I'm not going to talk about the TUCR arrays today, however. That's going to be part of a later webinar. So as far as numbers 1 and 2 are concerned, each has a different purpose, and so I'll devote the remainder of the talk to telling you the benefits of each. Okay, so how do we profile LncRNAs? Well, Arraystar has the Arraystar LncRNA microarray, expression microarrays, which is what I'm going to tell you about first. This is probably our best-selling product, the Arraystar LncRNA expression arrays. Now, this slide summarizes the important features of these arrays. The human version and mouse both come in an 8x60K format. In the case of the humans, there are probes for, there are 33,045 probes for LncRNAs and 30,000 probes for messenger RNAs. Similarly, on the mouse array, there are 31,423 probes for LncRNAs and 25,376 probes for messenger RNAs. And finally, in the rat array, which is a 4x44K, there are about 9,300 LncRNA probes and about 15,200 messenger RNA probes. So with our Arraystar LncRNA expression microarrays, not only do you get to profile all of the known LncRNAs in your project, but you get the added bonus of profiling all the known protein coding messenger RNAs as well, all in the same experiment. So why choose Arraystar for your LncRNA experiments? For one thing, we've designed our arrays using data from the most comprehensive genomic databases in the world, including RNADB, which is the non-coding RNA database from Australia, RefSeq from NCBI, the database called NRED, the non-coding RNA expression database, HINVDB, Ensemble, and UCSC. Also, our microarrays are designed in reference to some very important landmark papers that have come out in the past several years. Another reason Arraystar is the best choice for your LncRNA profiling needs is our probe design. First, we use highly specific 60-base long oligonucleotide probes that detect only targeted LncRNA transcripts as well as the protein-coding messenger RNAs. These probes are synthesized in situ for greater placement accuracy. Second, other companies' probes are typically placed singly or in clusters at the 3' end, and so can only detect a limited number of transcripts per gene. In contrast, Arraystar probes are targeted to exons or splice junctions, and so they can detect multiple spliced isoforms of each gene as well. Arraystar LncRNAs give you reliable, highly reproducible results. Figure 1, shown on the left, is a scatter plot between technical replicates in a typical experiment. In this figure, the tight scatter plot indicates that there is very little variation observed between the replicates. Over on the right in Figure 2 is a comparison between results of an Arraystar LncRNA microarray and qPCR results. And what we have found is that the results from an Arraystar microarray, shown in blue, show excellent agreement with that of quantitative real-time PCR, shown in red, in both the direction and magnitude of differential expression changes. Now for the last part of my talk, I want to convey that not only can we use microarrays to profile changes in LncRNA expression, but we can also use them to explore the mechanisms behind those changes in regulation. Which brings us back to this slide that I showed earlier. We know that LncRNA genes, like protein coding genes, are transcribed by RNA polymerase II, and so are regulated by the same control mechanisms: transcription factors, histone methylation, cytosine methylation, etc. Arraystar is keenly interested in developing products that profile the expression of LncRNAs. The expression arrays do just that. They tell us which LncRNAs are differentially expressed in a given tissue or experimental paradigm, as well as the magnitude and direction of that expression difference. But once we've learned which LncRNAs are differentially expressed, it would be natural to then ask what the causes of that differential expression are. So now I'm going to go over a recently introduced product from Arraystar, one that we are very excited about, the Arraystar LncRNA Promoter Microarray. Our new LncRNA Promoter Microarray is designed to profile all of those potential modifications of LncRNA promoters on a single chip. The Arraystar LncRNA Promoter Microarray is the first commercially available microarray devoted to LncRNA promoters. These arrays are useful either in a Chromatin IP experiment to identify promoter-binding proteins, or in a methylated DNA immunoprecipitation experiment to look for evidence of cytosine methylation. These arrays use specific, highly sensitive 50 to 70 base long probes on an ultra-high density 1 by 2.1 million feature array. Here's a brief scheme describing the two general ways in how the Arraystar LncRNA Promoter Microarray can be used. Suppose you're interested in profiling changes in the binding of your favorite human protein or some other DNA binding protein, denoted here as protein X, to a particular set of LncRNA promoters in different tissue types. Then you would do a chromatin immunoprecipitation experiment, cross-linking protein X to DNA, immunoprecipitating the chromatin with anti-protein X antibody, then hybridizing labeled precipitated DNA to the promoter array. In the second scenario, say you're interested in changes in LncRNA promoter CpG methylation between a hepatocellular carcinoma cell line treated with a drug and an untreated control. Then you would do a methylated DNA immunoprecipitation using anti-5-methylcytosine antibody to precipitate fragmented genomic DNA, which again is then labeled and hybridized to the array. Okay, this slide shows more detail about the features of the new LncRNA Promoter Microarray. Currently available for human, it comes on an ultra-high density 1 by 2.1 million feature format, as I mentioned before. On top of that, the coverage includes more than 28,000 LncRNA promoters, ranging from 3.5 kb upstream of the transcription start site to 1.25 kb downstream. In addition, the array features 30,215 messenger RNA promoters, again from minus 3.5 kb to plus 1.25 kb. Further, there are probes for 1,426 microRNA promoters, spanning approximately 15 kb of mature microRNA DNA. And on top of that, there are more than 27,000 CpG islands, more than 21,000 CpG island shores, K4, K27 bivalent regions, ultra-conserved regions, and Hox cluster regions. So, using the Arraystar LncRNA Promoter Array, you can answer many important and interesting biological questions all in a single, convenient, and powerful array. The Arraystar LncRNA microarrays are well suited to biomarker profiling in various diseases. As a case in point, NIH investigators Mark Zayats and Owen Rennert just recently published a paper where they used the Arraystar Human LncRNA version 2.0 array to profile LncRNAs and mRNAs in autism. The results shown here, summarized here, indicate that 82 LncRNAs are differentially regulated in the prefrontal cortex of autism patients, while 143 LncRNAs, different expression levels in the cerebellum. Again, this is an excellent example of using Arraystar's microarrays for the diagnosis, prognosis, and possible treatment of human disorders. Okay, so this brings me to the end of my talk, so I'd just like to briefly summarize. Today I told you that LncRNAs have recently emerged as major players in the regulation of gene expression for normal mammalian development, that LncRNAs have been linked to many diseases and so are potentially important biomarkers for diagnosis, prognosis, treatment, and drug discovery, that Arraystar has developed the industry-leading microarrays for profiling LncRNA expression, the Arraystar human and mouse LncRNA array version 2.0 and the Arraystar rat LncRNA array, and that we've also developed the first commercially available microarray for probing epigenetic modifications in non-coding and mRNA transcripts. Arraystar is committed to providing products that can help scientists analyze gene expression on a high-throughput scale. So I hope I've convinced you that Arraystar would be the best choice for your LncRNA profiling experiments. So that's about all I have to tell you about today. Again, I'd like to thank you all for taking the time to listen to my presentation. If you have any additional questions or would like to learn more about LncRNA profiling, feel free to ask me and to visit our website at www.arraystar.com. Okay, so that's the end of the talk, and now I'll take any questions. If anybody has any, I just need to pull up my control panel and look for them. Okay, does anybody have any questions? Like I said, if you do, feel free to type them into the question box in your control panel. Okay, well, it looks like nobody has any questions. Actually, somebody has their hand raised. That's Dr. Goswami. Would you like to ask your question? Okay, let's see. Oh, how did you design primers for validation? I actually don't know the answer to that question, unfortunately. The people who did the array design did that. I would have to find out from them. It's a very good question. So if you'd like to send me an email, or I could email you with the answer, that would be fine. Okay, does anybody else have any questions? Yes, Dr. Goswami, you have another question? Oh no, okay, that was the previous question. Okay, well, that seems to be about it. Anyway, I'd like to thank you all again for attending the webinar, and have a good day. Take care.